So hello students, welcome to my channel, and this is the third video that I am making for ISC Specimen question paper 2024 biology, and we are mainly discussing about here paper 1, that means theory paper. And already I made two videos on section A and section B. Please follow that video and don't forget to share and subscribe those videos. And now today in this video we'll be talking about section C and section D. So section C here, they will give you 7 questions, each will be carrying 3 marks, so total marks 21. So here first question in section C. The diagram given below shows various phases of the menstrual cycle in human. And then they have given this diagram where they, in one case, they are showing you the stages of the ovarian follicles, and the development of the eggs or the rupturing of the eggs, or what is going there inside the ovarian follicle, that is mainly indicating here in this diagram. Then in this level where it is stating hormones, that means different hormones levels are fluctuating in different phases of the menstrual cycle. So that's also showing at the middle ovulation, where that is mainly the egg just releasing out from the mature Graafian follicle after its bursting. So this diagram is mainly illustrating the menstrual cycle in a human. Okay, now related to this diagram, if you observe the question, the first question is that identify the plot lines that represent the following. So we have to identify the plot lines. So here progesterone. So which plot line is indicating progesterone hormone? So here the plot line 1. It is here they have given 4 plot lines, 1, 2, 3 and 4. So plot line 1 is mainly for progesterone. Another question in the same part, that is luteinizing hormone. So which one is showing here in the line, which one is indicating the luteinizing hormone? So here this line number 2, it is mainly for luteinizing hormone. Okay, so progesterone number 1 and luteinizing hormone, it is indicating number 2. Luteinizing hormone, why it is number 2? Because ovulation there, it is also known as the LH surge, that means LH at the peak level. So thus number 2 is mainly having the luteinizing hormone. Okay, now then next is the give any 2 differences between the menstrual cycle and the estrous cycle. That means what are the main differences between these 2 kinds of cycle. So let's see menstrual cycle. I have written 2 points as they asked for 2 differences. So menstrual cycle, it mainly occurs in females of primates, that means in monkeys, apes and human beings only. And estrous cycle, it mainly happens in females of non-primate mammals like cows, dogs, etc. So the first point is indicating where, for which organism these kind of cycles are mainly observing. Second is about the losses of blood. That means in case of menstrual cycle, blood loss occurs in the menstrual phase of this cycle. That means menstrual phase of this cycle means, actually in the menstrual cycle there are 4 phases. The first phase is the menstrual phase, and then there is the follicular phase, then there is the ovulation phase or ovulatory phase, and fourth is the luteal phase, where the corpus luteum mainly forms. So thus we can say in the menstrual cycle 4 phases are there. So the first phase of the menstrual cycle is menstrual phase. Okay, same name, menstrual phase. Under the menstrual cycle, thus it is written blood loss occurs in the initial phase of the menstrual cycle. But in case of estrous cycle, we will not see any kind of that means losing of the blood, so no blood loss in the estrous cycle. Okay, so this is about the difference. So this diagram, it is mainly given for 3 marks, so we discussed its question along with the answers. Now come to the second question. Now this question number 10, it's very confusing. I tried my best, but I have not written the answer, but I will discuss you here what they are actually trying to prove, what they want to ask. Actually, what is the main motto of this question and why they placed such kind of questions here. It is this diagram, it is mainly indicating the gel electrophoresis. Okay, and as it is mainly for DNA isolation, so we can say it is the agarose gel electrophoresis. Okay, because for protein gel electrophoresis, that is page or polyacrylamide gel electrophoresis. So the question is the three persons in an experiment named Kavya, David and Abdul had to cut a plasmid which is 650 base pair long to insert a gene of interest. All of them worked on this task individually. Okay. As per the protocol, a restriction enzyme was incubated for 2 hours with the plasmid. Okay. After some time, they loaded their sample onto the same agarose gel for electrophoresis. Given below is an image of the agarose gel where sample A belongs to Kavya. That means these bands it is placing in the sample A. These are actually the bands of Kavya for her that means sampling, and sample B, it is the bands are showing, that means it is for David, where he put his sample, and the samples are segregated in different density level, that means on the basis of the molecular weight and molecular length, we can also say, and in sample C it is for Abdul. So the question is that and this first line is mainly for marker, okay, which is, that means by seeing this marker we can say, and that means the samples of the bands are now in the which particular base pair level. So thus the marker is mainly used. Okay, and you can see the patterns, the thick size pattern, and gradually it's just slow, that means move down, and then at the last level, as it is 100 base pair, so it is thin. So the last one is very thin as compared to the topper one. Okay, so now if Abdul has performed the experiment correctly, that means Abdul means here sample C. Okay, if Abdul has performed the experiment correctly, what is the reason for the difference in the band pattern in Kavya's and David's sample? Now you can see this sample C, it is showing for Abdul's sample, and sample A and sample B, it is mainly showing for Kavya and David's sample. Now what is the main difference between the band patterns in sample A and sample B, that means in Kavya's sample and the David's sample? So according to me, it mainly happens due to two reasons. I have not written the answer, but I am just discussing here that this restriction engine, Kavya and David, they have not used same kind of restriction enzymes. This is my first point. The second is then if they use the restriction enzyme, it is not given in an equal amount. Okay, so thus this kind of variation mainly appeared because they have not used same kind of restriction enzyme. Okay, so as in sample B, that is mainly for David, so as the same restriction enzyme which sample A where Kavya put her sample, so as in sample B it is thick sized, that means restriction enzyme is not actually splicing the DNA desired gene part, so thus it is not segregated, because only after the cutting of the sample of the DNA from the plasmid, then only, or after the cutting of the portion of the gene, then only it is possible of its segregation in the gel electrophoresis. So as it is still attached here, so thus in this case we are not seeing any other band. That means restriction enzymes are not used here, we can say, or we can say the restriction enzyme which is used here for sample B, it is not actually working on that particular portion of the gene which they want, which one they actually want to place, or which one particularly they want to insert. This is my answer. Okay, and the thing is that according to Abdul, only a gene of the size of 200 base pair can be inserted into this plasmid, as this is the size of DNA that has been cut out from the plasmid. Is he correct? So actually in the plasmid the base pair is 650 base pair. So in 650 base pair plasmid, we can say Abdul can easily place 200 base pair, because from the plasmid out of 650 base pair, if we cut that particular 200 base pair length through the using of this restriction enzyme, there he can put 200 base pair of desired DNA part. So actually plasmid here it is using as a vector. Plasmid is the extra chromosomal DNA, and there what we are doing, we are using this plasmid as a vector. So we are putting 200 base pair desired gene after cutting that particular portion of the plasmid where the length is 200 base pair, and the same length of the base pair of the desired gene we are incorporating here, and thus this way we can introduce them as a vector so that we can easily transport that desired gene in any organism. Okay, we can introduce them in any organism. Okay, this way actually the genes are mainly transferring artificially. Okay, this is the part of recombinant DNA technology. Okay, so is he correct? So I can say yes, he is correct, and justify giving one reason, because as the plasmid size it is a 650 base pair, and as he used restriction enzyme, so restriction enzyme is mainly spliced on the desired part where 200 base pair length of the plasmid portion is present, and here he inserted that 200 base pair of the desired gene. So it is possible. Okay, I have not written the answer of this question because I told a lot of things here about this gel electrophoresis and why such kind of variation in the band patterns we mainly observe for these three samples, that means which placed by Kabir, David and Abdul. So thus I have not written anything, but I have discussed. Okay, next. It happened only for this question where I have not written the answers. But if we see the next question, here I put down the answers. Here I put it the answers here. The first, this is question number 11 of the section C. The figure given below shows a mushroom. This is the mushroom that is connected to the roots of another tree growing near it. You can see the in a picture in the diagram. It is written figure 10 because in the question paper it is figure number 10. So here you can see the root part of one large sized tree plant, and there in the same soil we can see the root portion or the spreading of the mushroom root portions there, which is just start moving around the root part of the large sized plant. So what kind of relationship it is actually, what kind of relationship is mainly building here? So identify the type of interaction between the mushroom and the roots of the tree. So here it is, we can say it is one kind of symbiotic or mutualistic relationship. Okay, now how we can say this is a mutualistic relationship? Actually they ask this question in the next part. Mention two advantages of this type of interaction. So in this case we can also say the advantage, or we can also say the reason why we can say it as a symbiotic relationship. So I have written that a mushroom either surround or penetrate the roots of trees and help the trees by conducting moisture and contain minerals to the tree's roots. Okay, so mushroom is providing what it is providing. It is after collecting the moisture and after collecting the minerals, so that minerals and moisture it is providing to the root part of the tree. Now in turn, the tree is what it is doing. It is transmitting the required nutrients for the fungal growth. Fungal means the mushroom growth here, and that can be absorbed by themselves. So this way these two organisms, the large sized plant and the mushroom, they are interacting. So their interaction, we can say it is a positive interaction. Okay, positively they are interacting. So both are beneficial here. So in this case we can easily say that it is the mutualistic or the symbiotic relationship where both the organisms are benefited. Move to question number 12. Next question of the section C. The homozygous Andalusian chicken exhibits black feathers, capital B, that means it is in a dominant form, and white feathers, that is WW, while the heterozygous bird has bluish feathers. Okay, so actually if we see the question, that means both the allelic pair in its dominant form. Okay, capital B, that is the dominant form. WW in the capital letter it is written, so it is also indicating the capital, that means the dominant form. So we can say that is indicating, that is mainly asking for codominance. That means where both the dominant pairs are expressing equally. Okay, now see the question. If two heterozygous Andalusian chickens are crossed, okay, if two heterozygous Andalusian chickens are crossed, what are the possible genotypic and phenotypic ratios of their offspring? Okay, So for both the cases, the genotypic and phenotypic ratios of their offspring, it will be 1:2:1. Okay, 1:2:1. We can say 1 it is for capital B, capital 2 it is for we can say BW, okay, and last is for WW, capital W, capital W, for that we can say. Okay, so this is indicating the ratios expressing in genotypically as well as phenotypically 1:2:1. Okay. Now next, what kind of dominance does the gene for feather color exhibit? As I already told, it is actually codominance, where both the dominant features are equally expressing. Now give a reason why we are saying that is a codominance, because both the genes of an allelomorphic pair express themselves equally in the heterozygous condition. Okay, so this is the nature of the codominancy. So thus this question is being asking and related to codominance. Next, name the category of microorganisms that aid in biogas production. So here methanogenic bacteria we are mainly using to form the biogas, and give one example of that category of the organism which we are using here. So one example I put here, that is methanobacterium. It is in the italic manner, that means this is the scientific name of the methanogenic bacteria. Okay, there are many methanogenic bacteria. I actually wrote one name in this is the scientific name of that organism. Okay, now next is the what are the advantages? That means mention any 4 benefits of biogas compared to other energy sources. Okay, so here that means 4 points we need to put. So first point is it is a storable form of energy which can be used when required. Okay, so stored form enough energy, and whenever it is required, that time we can use them. So this is one benefit. Another benefit is that it improves the sanitation and minimizes chances of spread of fecal pathogens. This way we can stop the spreading of the other fecal pathogens which may be harmful for the environment, harmful for the health issues. So that is the another benefit of this biogas production. Okay, it is an eco-friendly and pollution-free source of energy. Obviously, in this case, we are not using any such kind of things, or any such kind of elements are not emitting out from the system that can pollute or interrupt our normal environmental situation. So it is totally eco-friendly, and here there is no chance of that is contamination of any element with the air or to the water or in the soil. So it is totally pollution-free. Okay, and fourth benefit, we can say it is used as a fuel for cooking, lighting, and for power for running tube wells. Okay, so these are the 4 benefits of biogas as compared to other energy sources. Question number 14. Draw a well-leveled diagram of LS of Anatropous ovule. So here this is the diagram of Anatropous ovule, where we will get the chalazal end, and the opposite end is the micropylar end. Okay, and in that ovule we will get 3 kind of cell inside its embryo sac, antipodal cells, 3 antipodal cells, 1 secondary nucleus, that means where inside 2 colon nuclei are fused. Then this egg apparatus part, where 1 egg cell is there along with 2 synergies, those are assisting the eggs in its function. Okay, ovule, it has the outer coating that is called integument, and in that coating 2 layers are there. The outer coating is the outer integument, the inner coating is called the inner integument. Okay, and then this part is the funicle, and the food laden portion is the nucellus, which is given here. So this part should be labeled in your diagram, and this diagram is really very important for your exam. So this is LS of Anatropous ovule. LS means the longitudinal section. And here this diagram, it is mainly for mammalian testes. That is mainly the another LS, that is longitudinal section only. And in this diagram we will get the testicle part, that means which is covered by the scrotum, which is mainly present inside the scrotum of the scrotal sac. And here this portion where the important cells are present, we called it as seminiferous tubule. Seminiferous tubules, these are mainly present here. Okay, and they are separated, each seminiferous tubules are separated by a sheath or a membrane that is called the septum. Okay, septum, and then it's mainly released there in the part that is called retitestis. and from there through the efferent duct, which is called vasa efferentia, it start moving to the epididymis part. Now in the epididymis, this portion is the epididymis, where three zones are present. Caput epididymis, that means the head part of the epididymis, then body of the epididymis, and the last is the caudal epididymis, or the terminal part of the epididymis. From there, the epididymis, you can see the extension. There is the duct that is called the sperm duct of the vasa differentia or vas deferens. Okay, so this part should be labeled in your diagram. And this diagram is also very important. So this diagram, it is given from human reproduction, and this is given from the reproduction of flowering plants. Okay. Now next question for section C. So explain three different types of parasitism with one example each. Okay, so three different types. So I have written ectoparasitism, endoparasitism, pathogenic and non-pathogenic parasitism, holoparasitism, and hemiparasitism. Okay, ecto and endoparasitism, for that I have written the parasites which live on the surface of host's body are called ectoparasites, while the parasites which live inside the host body are called the endoparasites. That means those are present outside the surface of the host body. We can say ecto. Ecto means outside, and they are showing parasitic features. So parasites, so ectoparasites, and the parasites, those are situating inside the host body, sucking nutrition from the host body from inside, and then it is called the endoparasites. Second category is the pathogenic and non-pathogenic parasitism. Some of the parasites whose presence produces diseases in the host body, we can say they are pathogenic parasites. But some of the parasites, if their presence are not creating any kind of pathogenic situation, then in this case we will call them as non-pathogenic parasites. And non-pathogenic parasites are not harmful for the host body. Then holoparasitism and hemiparasitism. Holoparasites are totally dependent on the host for all their requirements. To fulfill all the requirement in their life cycle, they need to totally depend on the host body. Okay, so that is holoparasites. But hemiparasites, they receive only a part of their nourishment from the host body and manufacture rest part of their body. Okay, so that means holoparasites are truly parasites, fully dependent, but hemiparasites are partially dependent. Once they collected the nutrition from the host body, then they can use them, they can manufacture their food with the help of that taken nutrition. So it is hemiparasite. So I have written three kinds of parasitism nature. Move to section D, because I told already this video I am making mainly not only for one section but for section C and section D. Previous videos for section A and section B, two separate videos I made. See those videos, serially you go. First you see section A video, then you see section B video, and then after in this video, as I am covering both section and section C and D, so then after you can come and see that video. It will be more fruitful for you to complete your preparation. Okay, because to see the port pattern, you should know the specimen paper. You should aware about the specimen paper. What kind of question they will going to ask. By seeing this specimen paper, you can easily assume. Okay, so here Vallisneria is a submerged dioecious hydrophyte. The female flowers of Vallisneria reach the surface of water by their long stalk, while the male flowers are released onto the surface of water due to bursting of the inflorescence. Vallisneria is actually water pollinated flower. Water pollinated flower means hydrophilous flower. Okay, so now their question is that give any two characteristic feature of the pollen grains of hydrophilous flowers. So first, pollens are produced in large numbers. Huge numbers of pollens are mainly formed, which is economically not suitable for the plant body. But now we are not discussing about the advantage or the disadvantage. We are discussing about the characteristic feature. So this is one feature of the pollen grain. Another feature of the pollen grain is that pollens are protected from wetting. Wetting means that means as they are staying on the water, aquatic situation, so there is the chance of the damaging of the pollen grain due to the continuous sticking with the water body. So their surface, I'm talking about the pollen grain of the water pollinated flowers, their pollen grains are totally covered or surrounded by the mucilage layer. So it is called mucilaginous cover. So these two features are present on the pollen grains of hydrophilous flower. Next is state whether Vallisneria is adapted for autogamy or xenogamy. Autogamy means it is indicating self-pollination, and xenogamy is indicating cross-pollination. So here actually Vallisneria, for them the pollination is they are performing their pollination through the agent. Here the agent is water, so obviously it is xenogamy. There is no chance of autogamy. And why? Give one reason to justify your answer. This is the question for its statement. So transfer of pollen grains. They transfer their pollen grains from anther of a flower to the stigma of another flower through agent, and here the agent is water. Thus, we are considering Vallisneria as a xenogamic flower or cross-pollinated flower, where the pollinating agent is water. Give any 3 disadvantages of self-pollination. Okay, 3 disadvantage. Continuation of self-pollination results in weaker progeny. It will develop weak varieties continuously if it is happening. So through self-pollination, it is finally resulting in the formation of the variety having with weak, that means features. Weaker varieties will be formed. Weaker progenies will be formed through self-pollination. This is one disadvantage. Second disadvantage, the disease resistant power is also moving down day by day, okay, throughout the generation, because in the same way the same kind of genes are transferring, having same kind of features, having same kind of drawbacks. So it is the another that means drawback or the disadvantage of self-pollination. And third is there is no possibility of the formation of any new varieties, that is no variation. So third point is no variation, no new varieties are mainly formed through this self-pollination process. So these are the 3 disadvantages or drawbacks of the self-pollination. Next, a couple is unable to have children naturally, despite the woman's ability to ovulate and the man having a normal sperm. That means there is no problem in the female's reproductive system or the male reproductive system. Okay, and both their reproductive systems are that means successfully they are forming their gametes now, but they are unable to have children naturally. Now mention the 2 different types of abnormalities in sperm which cause infertility in males. Okay, that means there is a problem in the sperm. The sperm, that means sperms, it is actually present in a mixture where the accessory male reproductive glands, prostate, cowparts, and the seminal vesicles are releasing their fluids along with the sperm. That is, we can considering them as a fluid that is called semen. So in that semen has a low sperm count. This is another, that means abnormality. That means low sperm level, low sperm count in the semen. This is one abnormality in the sperm can cause the infertility of the males. Another is the abnormal sperm structure or poor sperm motility. The sperm structure is not fully developed, so it is abnormal development, or this poor sperm motility. The movement, it is also not perfect. So in this case, these are the 2 possibilities of the sperm features which mainly create the infertility in males. Suggest any 3 methods of assisted reproductive technology except GIFT, gamete intra-fallopian transfer, available to the couple to consider. So 3 methods. Number 1 is IVF ETT. The full form is in vitro fertilization embryo transfer technique. This is also called test tube baby. So sperm of male and ovum of female are induced to form zygote in a culture medium, and then embryo is transferred in the female genital tract for further development. This is one method. This is one assisted reproductive method. Another reproductive method is AIT. Full form is artificial insemination technique. So sperm of husband or donor male is artificially introduced into the vagina or uterus of the females. Okay. Another AST method is ICSI, that means intracytoplasmic sperm injection. So sperm of male is directly injected into ovum. Okay, so these are the three methods of assisted reproductive technology except GIFT. Okay, next is, what does the term biodiversity mean? And explain four major causes of loss of biodiversity due to human activities. So biodiversity refers to the variety and variability among living organisms and the ecological complexes in a given area. It represents sum total of various types of microbes, plants, and animals present in an ecosystem in a given area. This is the definition of biodiversity. Now, four causes of depletion of the biodiversity due to human interference. So number one is habitat destruction and fragmentation. Second cause, overexploitation or improper resource management. Third, alien species invasions. And fourth, coextinctions. So there are four major causes of biodiversity losses: habitat destruction and fragmentation, overexploitation or improper resource management, alien species invasions, and co-extinctions. Last question. It is given from molecular basis of inheritance. A research group studied various aspects of a particular region in their gene of interest and its muted versions. Shown below is a part of the DNA sequence by where studying 5' to 3'. These are the sequence. Now come to the question, and they have given this information about the genetic code and which particular amino acid is formed from which codon. So what is the DNA sequence complementary to the above sequence? So here they have given 5' to 3' sequence. That means its complementary sequence will be from 3' to 5'. So here I have written 3', and this is the sequence that is totally complementary to the sequence which they have given. So that was 5' to 3', and now this one I have written its complementary strand as 3' to 5' series. Okay, 3' to 5' direction. Okay, so this is the sequence complementary base pairs, because two DNA strands are parallel, but they are anti-parallel. Okay, so thus we need to make in such way. Write the sequence of mRNA transcribed by this segment of DNA. Okay, now we should know on which template strand, on which strand of the DNA is mainly using as a template form to form the mRNA. That means for transcription process. So actually 3' to 5' strand is mainly using as a template strand of the DNA, and by sequencing, that means by taking that strand mRNA which is producing now as 5' to 3' direction. So this is the sequence which will be formed: AUG, UUG, SCA, UCA, UCC, AGC, UGU. So you can see this 5' to 3', it is totally equal to this sequence, but one difference is that instead of T there will be U, because in case of mRNA thymine is absent, and in place of it uracil is present. So thymine is present for DNA and thymine is absent for RNA. So in case of RNA, thymine is not there. Instead of it, uracil is present. So on the place of T, we need to write U. So if you see that, it is same only. Okay. So thus, as it is same 5' to 3' strand of the DNA, as it is same as the mRNA, except obviously where the T is not there, U is there. So thus this 5' to 3' strand is called coding strand. This is called coding strand, and this sequence on which the mRNA will form, and that is known as the template strand or anticodon strand. Write the amino acid sequence coded by the mRNA sequence using the genetic code shown in the figure given above. So it is written, that means this is 5' to 3', and according to the codon, the particular amino acid is written, and that one I have written by picking the information from here, the information which is given here for different amino acids, which particular codons are responsible and which particular codons are mainly coding for the formation of the amino acids. So that one you will get it from here, and by seeing this codon we can form this amino acid sequence. A mutant of this gene contains A instead of C in the 11th position in the given DNA sequence. What will be the amino acid sequence coded by this mutant gene? So TCA, see, just move through this diagram. TCA, here it is. If you count it, TCA, we will get it is in the 11th number position. If you count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So this is C, which is in 11th number position. So what they told that gene contains A instead of C. Actually C was there, but now it is A. So they are talking about the 11th position. So that means the mutation happens. Okay, mutation happens. That is also called point mutation, because one particular nucleotide base pair, one particular nitrogenous base pair, it is replaced by someone. That is called the point mutation. So that happens in the 11th position. So due to the change in the 11th position, so that means that codon pattern is also changed. So what will be the amino acid sequence coded by that mutant gene? So previously it was TCA, where C is in the 11th position. So due to mutation now it is turning into UAA, because now we are talking about mRNA here. Okay, in the DNA level, if the gene mutation happens, so that will be reflecting in the formation of the mRNA level. And I told that it will be forming the complementary base sequence. So UAA will form, and that UAA will not be coding for any amino acid, because it will be coding for that means we can say this UAA, it is the stop codon or the termination codon. Okay, that is one nonsense codon. It is not coding for any particular amino acid. So that mutation in the 11th position, where C is replaced by A, in this case the codon which will be formed, transcribed, that will not be able to code for any particular amino acid. This is the problem which we will be going to face due to the mutation if it happens in that case. Now, what type of mutation would the change in nucleotide referred to in subpart 1.4 lead to, and justify by giving one reason. So as the nonsense codon is mainly formed through this mutation, thus this mutation is called nonsense mutation. So as this mutation produces nonsense on termination of the stop codon UAA, so thus this mutation we can say it is the nonsense mutation. So these are the questions which they have given. This is the last 18th number question. Okay, so thank you for watching this video. If you like this video, don't forget to share, subscribe and like it. Okay, thank you and have a good day.